All right, welcome to the first Ethel Browne Harvey Postdoctoral Seminar of the new season. I'm Ray from Harvard Medical School, and I will be moderating today with Dr. Taylor Medwig-Kinney from the University of North Carolina at Chapel Hill. We have an exciting lineup of speakers in the 2025 to 26 season. We hope you will join us throughout the year. Today, Dr. Sarah Colijn from Washington University in St. Louis and Dr. Shyama Nandakumar from the University of Pittsburgh will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A. Please enter your questions in the Zoom Q&A box. Our first speaker, Dr. Sarah Colijn, is a postdoctoral fellow in the lab of Amber Stratman at Washington University in St. Louis, focusing on the functions of cilia in the embryonic development of vasculature. Sarah did her undergrad degree in biochemistry and PhD in cell biology at the University of Oklahoma, also studying vascular biology. She finds the cardiovascular system fascinating because it is the first organ system to grow in the embryo, and it is integrated into and essential for the function of all the other organ systems. During her PhD, Sarah elucidated the mechanisms of RIPK3, a key enzyme in cell necroptosis that has an unexpected protective role against atherosclerosis in mice. She also revealed the critical roles of its epigenetic regulator in early vascular development. During her postdoc training, Sarah switched to the zebrafish model and took advantage of its transparency to study the early development of endothelial cells. Sarah has won many awards, including the American Heart Association Fellowship and the NIH K99 Award. Sarah, the floor is yours.
Thank you, Ray. I am very honored to be the first speaker in this seminar series, so let's get to it. So this story that I'm going to tell you today begins with the cilium, and in particular, how cilia regulate vascular development. That is, in a nutshell, the topic of my K99 RO award. So I'm going to tell you today about one direction that this topic of study has led me to. So what you need to know about cilia for this talk is that cilia are microtubule-based structures. They are cell protrusions. The base of the cilium is formed from one of the centrioles, which is also a microtubule-based structure. And these centrioles form a microtubule organizing center, or MTOC, for the cell. And defects in cilia components can manifest as numerous syndromes in humans. But for example, congenital heart disease patients often exhibit ciliary dysfunction. So CHDs are the most common type of birth defect, affecting about 1% of babies in the US and nearly 2% of babies worldwide. And there are many types of CHD, many anatomical variations of each. And 20 to 30% of cases have known etiology, but most cases have no known cause. It's believed that around 90% of cases could be attributed in part to genetic causes, but we just don't know what those genes are yet. So Peter Jin, one of our collaborators here at WashU, has access to data from the Pediatric Cardiac Genomics Consortium. And this consortium includes samples from CHD patients plus their parents. And these trios samples undergo whole exome, genome, and RNA sequencing. And from these data sets, Peter has thus far identified rare variants that associate with CHD. So knowing that ciliopathies can often cause congenital heart defects, we collectively mined the data for genes that have known association with cilia and found that this centriolar gene, WDR90, was decently represented in the data set. These 10 patients have rare variants in WDR90 that are likely causative mutations for CHD. And the phenotypes in these patients are varied, as there are many variations of CHD, which is not unusual, as CHD can be a very polygenic disease. So while these data do not tell us that these variants in WDR90 alone cause CHD, they certainly raise it as a gene of interest. Furthermore, 4 of these patients actually share the same variant. And when I compare the incidence of this variant in the consortium population compared to variant alleles reported in the general population from the NOMAD server, I find that this Q975X variant is significantly enriched in the CHD cohort. This further suggests that this variant may be causative for disease. What's extra interesting here, and really just some icing on the cake for you, is that these 4 patients all claim Puerto Rican ancestry, which may indicate that this Q975X variant is a founder variant in that population. So what is WDR90? WDR90 has a DUF domain, which stands for Domain of Unknown Function, and it has 4 beta propellers. And there are basically 2 papers out there that look at WDR90 function. They're both elegant papers from the same lab. And one of the papers shows by expansion microscopy that WDR90 is a centriolar wall protein. So these authors also showed that the N-terminal domain, that DUF domain of WDR90, is sufficient to retain the microtubule binding function of WDR90. This other paper shows that WDR90 at the centriole of the cilium, and when you knock down WDR90 using siRNA, the cilium no longer forms. So based on these data, WDR90 binds microtubules and is important for cilia assembly. So we acquired an uncharacterized WDR90 mutant zebrafish line from Zerk. It has a premature stop codon in exon 9, which is right after the DUF domain. So we bred the hets together to analyze embryonic phenotypes, but by 4 days post-fertilization, I saw no overt phenotypes in mutants versus clutchmate controls. And mutants were present at Mendelian ratios at 1 week post-fertilization. This is in contrast to other cilia mutants, which in general do not survive to the first week and have this distinct curved tail. So this made me think that, if WDR90 is important for cilia biogenesis, then maybe this is not a full knockout. This means that, if a protein product is still being made, that it would just be the DUF domain. And so to get at answering whether that may be occurring, I looked at transcript levels of WDR90 in these fish, and there's not even a 50% reduction in the mutants, suggesting that these transcripts are not fully undergoing nonsense-mediated decay, and suggesting that there is a truncated product being made. So as the previous studies had shown, this DUF domain can still bind microtubules, so maybe cilia can still form in these fish. And indeed, when I used an RL13B GFP transgenic line to visualize cilia, they appear to be able to form in the WDR90 mutants. All of these little green dots are cilia within the brain of the embryos. So this doesn't mean that ciliary signaling pathways are completely normal, but it does suggest that there are not severe ciliary defects in these mutants. Then I focused on the embryonic hearts, which are undergoing major developmental steps within that first week. So only about 20%, less than 20% of mutant hearts displayed some sort of defect. And while this was not a high number, it was significant compared to wild-type controls. These defects were also varied. So for example, laterality defects, like this shown in this image with the atrium and ventricle sitting atop of each other instead of lateral. And some had maybe just general delayed development. So I couldn't pinpoint one type of phenotype, but from this I suspected that any heart phenotypes associated with mutations in WDR90 in fish were either not fully penetrant or could be progressive. So I then let these fish grow to adulthood, and I found that the mutants were underrepresented by 3 months of age, indicating that the mutation caused a partial lethality after the embryonic stage. You can see that the surviving adults look grossly normal compared to clutchmate controls. So why are they not surviving as well as the controls? We then took a look at their adult hearts, and we saw a lot of variability in mutant heart size compared to clutchmate wild-type fish. In this example, the heart is very small in the mutant, and this vessel in the back here, which we think is the hepatic portal vein, has a coarctation, which is another phenotype associated with congenital heart defects. So given the variability, again, it was hard to determine exactly what was going on with these hearts. So we sent them off for RNA sequencing, and our results showed overwhelming upregulation of immune responses and pathways. And this is often indicative of heart failure. So does this variability in size and this inflammation have any noticeable effect on the actual fish? To answer that, we performed behavior assays. And you can see very clearly here in the in no flow conditions that the mutants are barely moving around, as if, they're conserving energy. And some of these mutants, if you look closely, are overgilling, which is equivalent to gasping for air. We also tried an endurance assay where the fish are subjected to increasing flow speeds until they become exhausted and can no longer swim. And younger mutants were able to keep up fairly well with controls, but by 12 months of age, when they may be in the midst of heart failure, the mutants, and even hets to an extent for males and females, are not able to swim as long as wild type. So this is more evidence that the heart function is impaired. So we took a step back and performed RNA sequencing on a whole 2-week-old juvenile fish. We chose 2 weeks because it was a time point after major heart development stages, but prior to the adult mutant lethality, so when there was no obvious difference still between controls and mutants. And we found some interesting groups in the pathway analysis. First, cell junctions were well represented in the upregulated pathways, as are cytoskeletal components like actin, extracellular matrix, and vesicles. In the downregulated pathways, some cilia genes did pop up, but they were related to eyes mostly and photoreceptors specifically. So of the actin-associated genes, there were actin-binding proteins, and let me summarize. We saw catenins, integrins, myosins, and in the junctional gene list, there were cadherins, claudins, collagens. So despite WDR90 being characterized as a centriolar protein, centrioles, cilia, microtubules, they were not major hits in this pathway analysis. But I got caught for a moment on the pathways showing downregulation of the few cilia genes, all related to the eyes and photoreceptors, like I said, all listed here. And it occurred to me that, do these fish have impaired vision? Are they less active because they can't see as well? So am I attributing an eye phenotype to a heart defect? So we performed a visual acuity assay on these fish. And this assay uses screens with moving lines at various speeds and thicknesses. Then it tracks the eye movements of the fish as they track the moving lines. And so what these graphs below here show is that there's actually no difference between eye movements in controls and mutants. So vision is not impaired, which again, points us back to the hearts as being the main reason for the decreased activity and endurance in the adult fish. So let's briefly think about the cell types that make up the heart. On the most basic level, the heart is made of cardiomyocytes, endothelial cells, smooth muscle cells, and fibroblasts. And just looking at WDR90 transcript levels in a few of these cultured cell types, I saw that endothelial cells and HUVECs particularly have high basal levels of WDR90 compared to like smooth muscle cells or fibroblasts. So I performed WDR90 immunostaining in HUVECs, and the first thing I asked was, is it localizing with centrioles at all? Because that's where it's known to be located. And when I zoom in on centrioles, yes, it is localizing there. But I noticed that it also appeared to be showing up at the cell borders, as the RNA-seq suggested it would be, as the pathway analysis identified junction and adhesion-associated genes. And there's also a lot of intracellular WDR90 here that looked intriguing. So to assess what was happening on the intracellular spots of WDR90 first, and to see whether it was binding to microtubules outside of the centrioles, I stained for microtubule minus and plus end markers. And the minus end is often situated and stabilized in the MTOC, that microtubule organizing center, and new tubulin dimers are added to the plus end. So one marker of microtubule plus end is EB1, and I immunostained for EB1 and WDR90. They do not colocalize. Then I immunostained for the non-centrosomal minus end marker CAMSAP3, and while not every WDR90 dot colocalizes with CAMSAP3 and vice versa, I do see some actual colocalization of the two, suggesting that WDR90 may be found as some microtubule minus ends. However, the regions where I saw the most distinct colocalization of CAMSAP3 and WDR90 seem to be at the cell borders. And since adherens junctions at endothelial cell borders utilize V-cadherin, I stained for V-cadherin to help just highlight the borders. But not only are WDR90 and CAMSAP3 at the borders, but they clearly colocalize specifically with V-cadherin. And to validate this WDR90 localization another way, we performed live imaging with a GFP-tagged WDR90, along with constructs to label actin, tubulin, and EB3, the microtubule plus N marker. And using RPE cells, which are an epithelial cell type that transfect way more efficiently than HUVECs, WDR90 can be found at the microtubule organizing center, where the centrioles are, and elsewhere it looks punctate and moves around a bit sporadically, which can be seen in this tracking graph here. It's like it's jumping around from microtubule to microtubule, suggesting that it's utilizing microtubules maybe as a mode of transportation as opposed to assisting directly in microtubule assembly, like a vesicle. At the regions where these cells could form adherens junctions, we also see WDR90 localizing at the junction region, which recapitulates the immunostaining. And what stood out to me about this localization at adherens junctions is that while microtubules have peripheral roles in regulating adherens junctions, the main cytoskeletal component there is actin. And actin-associated genes were misregulated in the WDR90 zebrafish mutants. So is WDR90 interacting with actin too? And I did observe intense WDR90 signal on actin filaments with V-cadherin. However, WDR90 can be seen on actin filaments by itself without V-cadherin, just at lower levels, suggesting it can be there, independently of V-cadherin. I then performed some super resolution imaging using STED microscopy, and through STED, we were able to achieve about 50 nanometer resolution. And WDR90 is in yellow, tubulin in magenta, actin is in cyan. And you can see in C and D here that WDR90 is colocalizing directly with tubulin. And in E and F, it is also colocalizing with intracellular actin filaments. So these data suggest that WDR90 can interact in some way with both microtubules and actin. So how are the patient variants affecting WDR90 localization? I created these GFP-tagged truncated versions of WDR90. This one is equivalent to our zebrafish allele, and the Q975X variant is the variant that was enriched in the PCGC. It has a premature stop near the end of the second beta propeller. And I made a version without the last two beta propellers to mimic how the patient variant may be expressed. So again, full length WDR90 is at the MTOC. It's somewhat vesicular and at the cell junctions. The N-terminus, though, which was already shown to have microtubule binding capabilities, colocalizes very smoothly with tubulin. Notice also in these images how the microtubules in the cell are very curved, which suggests that these microtubules are overly stabilized. Next, we looked at the patient variant and the similarly truncated form, and also saw that in both cases, it could only localize to tubulin. And unlike that first domain just by itself, this one looked very patchy on the microtubules. And the microtubules, again, looked very curvy and overstabilized. So this is what that overstabilization looks like when between wild type and the patient variant when you trace the microtubules. So with such severe effects on the cytoskeleton, this human variant likely does have functional defects. So to figure out which domain of WDR90 could be important for localization at adherens junctions or for actin binding, I created these GFP-tagged truncated versions of WDR90. And a couple of these constructs just expressed as a nonspecific cytoplasmic haze, but I'll show you data for the rest of them. So again, full length, MTOC, vesicles, and cell junction. Just the N-terminus is on microtubules. But the four beta propellers by themselves mimic the wild-type full-length version at the MTOC and the junctions. And the last two beta propellers are sufficient as well for just localizing to the MTOC and the junctions. Finally, the third beta propeller by itself colocalized solely with actin and was particularly bright on the ends of stress fibers. So the third beta propeller is an actin-specific binding domain. But is it interacting with actin directly? Is it influencing actin dynamics? For that, we turn to a collaborator to help with protein purification and turf microscopy. So in this assay, our collaborator Jess Henty-Radilla, purified some of the WDR90 fragments that I designed, and she combined them with actin monomers, then imaged and quantified the nucleation and growth of actin filaments. Actin can slowly polymerize on its own. The DUF domain alone appears to inhibit nucleation, and the fourth domain does nothing. But the 4 beta propellers, the first 2 and the last 2, did increase actin filament formation. So this is a very clean assay without any other proteins. And this suggests that more than just the 3rd beta propeller can interact with actin, though perhaps it's a 3rd that has higher affinity for actin in live cell imaging experiments. So she's still working on analyzing the 3rd beta propeller alone, but this shows that there can be direct functional interaction with actin and WDR90. Then Jess performed the same experiment with tubulin and WDR90, and these data are more preliminary, so there are fewer fragments that have been tested. But what we can see here is that the DUF domain and the beta propellers actually prevent microtubule formation by decreasing the elongation rate and microtubule length. So this is interesting because since mutant WDR90 overstabilizes microtubules, my first thought would be that, thank you, that it assists in microtubule assembly. However, this suggests more of an inhibition of like overall microtubule dynamics, both through preventing the assembly and disassembly of microtubules. So this is our current working model. Is WDR90 interacting with both microtubules and actin simultaneously? Well, we think it can. We confirmed that the DUF domain is very specific to microtubules and that the 4 beta propellers, but specifically the 3rd, can bind actin. So it would appear that WDR90 may have that ability to bridge cytoskeletons. The Q975X variant, on the other hand, has lost the actin-specific binding domain and presumably can only now bind to microtubules. Same with the zebrafish mutant, leading to microtubule overstabilization and WDR90 mislocalization. So this disruption of cytoskeletal dynamics could be the reason the heart begins to function poorly in the fish, perhaps resulting from an arrhythmia that eventually leads to heart failure, but that we are still looking into. So that is all I have for you today. I would like to thank my mentor for being an amazing mentor. And this has been a highly collaborative project, and I really want to give a shout out to a grad student in our department, DeHaven, who is magic with live cell imaging experiments and has provided most of the live cell imaging that you saw in my presentation today. So I'm ready for any questions.
Thank you, Sarah, for the great talk. Please enter your questions in the Zoom Q&A box, and I will read them. So I'd like to start with a question I have. So your WDR mutant has a very dramatic effect in the RPE cells by skewing all the microtubules. And I was surprised that the mutant zebrafish can still develop. Do you think that mutation affect all cell types equally, or it's the RPE cells that is very susceptible to this mutation?
So I did check just by immunostaining in human cell lines that WDR90 co-localizes to the borders in, smooth muscle cells and fibroblasts as well. So its ability to just, or the fact that the mutant only goes to the microtubules, I think could affect a lot more cell types than just epithelial cell types. There is one thing I haven't shared about the fish yet because I didn't have time, but I find that they actually have a little bit of genomic instability. So the progeny do not have the Mendelian ratios that I always expect when I cross homozygous mutants with, like, say, with wild type. You would expect then that all of the progeny would be hets, but I see wild type and mutants and hets. So I think that it's definitely affecting, the gonads and those cells as well.
Okay, great. We have a question from Celeste Berg. Does WDR90 interact with formins or other microtubule actin proteins?
So I don't know the answer to that question, specifically those microtubule binding proteins, but it is localized near CAMSAP3, which is a microtubule minus N marker. And by STED, we get fairly good resolution, so you can see whether they're directly interacting or just kind of sitting next to each other. And CAMSAP3 and WDR90 are not directly interacting, as we can see by STED. There may be some proteins in between. So what WDR90 may be directly interacting with other than microtubules or actin, we're not sure yet.
Cool. A question from Myler Lee. Awesome work. I'm curious how the zebrafish WDR90 allele with such an early premature stop isn't getting targeted by NMD. Any evidence for alternative splicing, for example?
So the fish has 2 splice variants of WDR90. And I should also mention that, fish often have duplicated genomes, but there's actually only 1 copy of WDR90, which is helpful for our study. But of the 2 splice variants of WDR90, one's the full length, another is a somewhat shorter version, but they both start and have the DEF domain and the first 2 beta propellers. So I don't think that premature stop that's right after the DEF domain would, promotes splicing of another variant because it would affect both, if that makes sense.
Yeah, a kind of related question from Gage Crump. Have you considered making a full locus deletion allele to control for transcriptional adaptation and role of possible truncated protein products?
Yeah, so we can't remove WDR90 completely, like we can't take out the complete locus because it is actually situated in the promoter region of another extremely important cilia gene going the other direction. So if we took out WDR90, we'd completely affect this other gene and they would absolutely die early and we wouldn't be able to tell, well, is it that gene or is it WDR90? But I'm working on trying some morpholinos to try to this doesn't exactly answer the question, but I am trying some morpholinos or a little bit more targeted CRISPR to try to get rid of WDR90 without removing the whole locus and, getting rid of maybe little truncated forms of it and just being a complete null.
There's a question from Taylor Medwig-Kinney. Do you have any plans to create conditional mutations or conditional rescues in the specific heart cell types?
So ideally, those experiments would happen, pretty easily in fish. If you want to rescue a phenotype, you could, put your wild type protein back in pretty easily at the 1 cell stage. It's a pretty simple experiment in fish. However, our phenotype is very long term. Like it's, 12 months they have heart failure. And at the early stages, I'm unsure what we're exactly seeing if there's something that we can truly rescue, because when you inject, at the 1 cell stage, you're getting a mosaic expression, the whole fish is not going to be affected. So it's something I've been thinking about, but technically it would be difficult given the phenotypes that we have.
Cool. Celeste Berg has another question. I think you might have partly addressed that. So the question is, if you created a true null in fish, do you expect them to be embryonically lethal, or can other proteins substitute for WDR90?
I suspect that it will be embryonic lethal. Part of the reason I think that is I for a while was trying to knock down WDR90 in just cell lines using CRISPR or siRNA, and I could never recover cells. And so next, my plan for that, the cells, is to use p53 null cells to keep them alive so I can knock down WDR90. But I suspect, given the fact that I couldn't keep the cells alive, that the fish would probably also perish early on.
Okay, great. Thank you very much, Sarah. Let's hand over to Taylor for the second talk.
It's my pleasure to introduce our second speaker, Dr. Shyama Nandakumar, a postdoctoral researcher at the University of Pittsburgh in Dr. Deepika Vasudevan's laboratory. Dr. Nandakumar earned her PhD in molecular, cellular, and developmental biology at the University of Michigan, where she worked with Dr. Laura Batista. Following her doctoral work, she pursued postdoctoral research at Cornell University with Dr. Nicholas Buchan before arriving at Pittsburgh. Dr. Nandakumar's research focuses on fundamental questions of cell biology and stress response using Drosophila model systems. As a graduate student, she discovered that polyploidy protects against DNA damage-induced cell death in the adult Drosophila brain, revealing important insights into neurobiology and aging. With the help of an undergraduate student, she also created Polyploidy Atlas, a curated, searchable, and sortable list of instances of polyploidy across metazoa. This resource can be accessed at www.polyploidy.dev. Her current research investigates stress response mechanisms, which she will be telling us about today. And be sure to keep an eye out for several exciting manuscripts from her work that are on the way. Beyond her research, Dr. Nandakumar is deeply committed to the scientific community, publishing experimental protocols, serving on Flyboard, helping to organize scientific meetings, and mentoring the next generation of scientists. Please join me in welcoming Dr. Nandakumar.
Thank you, Taylor. Good afternoon, everybody. I'm excited to be here today. I thank the organizers for giving me this opportunity to present my ongoing postdoctoral work. So my work in the Vasudevan lab at Pitt focuses on addressing two key questions, and the first of which I'm going to talk to you about today, which is how do cells within a tissue coordinate their stress response? So something we like to think about a lot in the Vasudevan lab, otherwise known as the fly stress lab, is how metabolic tissues such as the liver, the adipose, or the pancreas, which all share high biosynthetic burdens, endure constitutive stresses such as the unfolded protein response or ER or endoplasmic reticulum stress, while maintaining their functions. And as our lab's name suggests, we rely very heavily on the small but mighty Drosophila melanogaster, and we use its fat body as our model tissue. So for those of you who don't think about the fat body or flies every day, let me introduce this tissue to you. The fat body is the liver and adipose-like tissue in Drosophila, shown here in beige, that facilitates the exponential growth, which is characteristic of early Drosophila larval development. So flies develop from embryos to the last larval stage in less than 5 days, and this growth encompasses approximately twofold increase in body mass. And most of this is facilitated by the fat body, which plays multiple important secretory, endocrine, innate immune, and nutrient storage functions. And this is also a tissue that endures very high levels of basal stress, and it's easy to dissect, so it makes it a very good model tissue for us to study stress in. So some of our early studies interrogating the heterogeneity in the expression of a cell stress reporter in the fat body revealed an interesting pattern where, some cells show very high levels of the stress reporter, the details of which are here and I'm happy to talk about later. And some cells show lower levels of stress response reporter. And I was recruited to the lab partially to study this, but I became interested in a slightly different phenomenon, which is that the cells that show high and low levels of stress reporter expression always appeared to be in pairs and in neighboring cells. And so this pattern sort of reminded me of we just looked at other literature, and when we do mosaic clonal expression of transgenes in our own hands, or when we look at literature, when people express transgenes in a mosaic pattern across the fat body, I saw this pattern be recapitulated. And this reminded me of ring canals, which is a structure I'm going to introduce to you. Ring canals, known otherwise as septin rings, plasmodesmata, or intercellular bridges in other model or non-model organisms, are all names for essentially a structure, a conduit that's formed when cells undergo cytokinesis, but incompletely. So cells dividing don't completely divide and stabilize their midbody to give rise to an open channel that allows for transfer sometimes of intracellular contents. In Drosophila invertebrates, ring canals or intercellular bridges have been extremely well studied, and in the context of the germline for facilitating cytoplasmic organelle transport and for development of gametes. However, the presence and function of these structures in somatic tissues has been less well studied, and so we thought this would be an interesting thing to investigate in the fat body. So, does the fat body have ring canals? Because cells can be paired due to other reasons. I acquired a fly line that expresses a GFP, which should localize to both nuclei and ring canals. It's called Pavarotti GFP. And sure enough, we saw several small punctae on cell membranes across the fat body. And this resulted in a tissue geometry where each adipocyte was paired to exactly one neighboring cell through these Pavarotti GFP positive punctae. Now, Pavarotti GFP can also mark mid-bodies and not necessarily open ring canals. And so the question was, how are they ring canals? Are they open? And if so, how big are they? And the answer to that question is fat body ring canals are actually very, very small. We needed to perform super resolution STED microscopy to even verify that they are open. And the image on top here shows a confocal micrograph at 100x magnification. And this is a STED deconvolved image, STED super resolved and deconvolved image showing that this channel is indeed open with extremely small dimensions. So I performed this sort of super resolution imaging followed by measurement of the dimensions throughout larval development. And interestingly, unlike in the germline, the Drosophila fat body ring canals do not grow in inner or outer circumference, even as the cells themselves grow exponentially in both nuclear DNA content and cell size. So another thing that I saw was that the earliest larval stages always showed the presence of ring canals in the fat body, implying that these are formed before the exponential growth of this tissue. In fact, what we think is happening is that the last mitotic cell cycle of the mesodermal progenitors that give rise to this fat body culminates in an incomplete cytokinesis that gives rise to paired adipocytes that then individually undergo approximately 7 rounds of DNA replication, giving rise to adipocytes that have that are paired, that have 2 nuclei, each with 250 copies of the genome. And this phenomenon is quite similar to accessory glands in the prostate of the Drosophila. So if you want to check out this paper by a former colleague of mine, please do. Okay, so my next question became, do ring canal paired cells actively share cytoplasm? And to do this, we developed a photo bleaching recovery, fluorescence recovery after photo bleaching method, where we dissect out fat bodies and embed them in a low melting agarose and perform photo bleaching of individual cells. And then we can measure the change in fluorescence of the bleached cell and its several neighboring cells that are either connected through a ring canal or not. And so some of the images I'm going to show you, the cell that I'm bleaching will be outlined in a solid orange line. The sister cell, or the cell that it's paired to through a ring canal, is also in orange, but a dotted line. And then a neighboring cell that it's in contact with, but not connected through a ring canal, is shown outlined in green. And then I'm just going to play this video. This is the bleached cell. You might not have seen it, like the recovery is that rapid. So bleach, recovery. However, so this is in a matter of like 4.5 minutes. When I bleach both cells that are paired through a ring canal, over the course of my imaging, we never see a recovery of fluorescence, suggesting that whatever recovery I showed you in the previous video is a result of potential intercellular transfer through ring canals and not de novo synthesis of GFP protein. And this data is just quantified here as absolute GFP intensity or the recovery in each of the cells that are labeled. So now with some confidence, knowing that the photobleaching recovery paradigm and timescale that I'm using only represents possible transfer through ring canals, we can use the mighty Drosophila toolkit and ask, okay, what goes through these ring canals using a number of different fluorescently tagged reporter proteins. So in this video, the fat body cells are overexpressing an ER-tagged GFP, and within a matter of less than 6 minutes, we see a recovery of up to about 40% of fluorescence. And so let me remind you, if a cell is transferring GFP from this cell to this cell, the maximum recovery that you can see is only 50% of pre-bleaching levels of GFP. So the fact that we see 40% or 30% recovery within a few minutes is quite impressive levels of cytoplasmic streaming between these cells. I also did this with Golgi tagged GFP, and Golgi GFP passes through the ring canals just as fast, if not faster, suggesting that these two organella components are probably shared by two cells connected through ring canals. I don't have the time to go through all of the additional data, so I'm just going to tell you that I've checked mitochondrial GFP, and I've tried to see whether lipid droplets go through ring canals, and neither of these appear to be transported through these ring canals. And this tells us that the transport facilitated by these very small ring canals is not indiscriminate, but highly specific, although rapid. Right? So as any geneticist would do, the next question became, what happens when we disrupt these ring canals? And to do this, we turned to the literature and found that depletion of Kelch, which is an E3 ubiquitin ligase that is known to be required for the maintenance of ring canal size in the germline, is something that we could knock down. So this is an image from the Cooley lab showing a mutant of Kelch that shows a much smaller ring canal opening. And I just want to show you here that this is a nucleus with roughly the same amount of DNA connected to another nucleus nurse cell of the same amount of DNA as fat body cells, but the ring canals are about 2 orders of magnitude larger in the germline than they are in the fat bodies. So we knew that based on data sets that we already have in the lab, that Kelch is expressed in the larval fat body. So we turned to depleting it. And when I do this, either by fat body specific knockdown or using a heterozygous Kelch mutant, we see that both of them result in a reduced inner circumference of the ring canals, similar to the germline. So now we have a great way to reduce the size of the ring canal. We can next ask, okay, how does this impact cytoplasmic transport and other components, other aspects of larval fat physiology? So I'm going to show you here 2 FRAP videos of a control RNAi, where you can see rapid recovery of fluorescent protein, whereas when we knock down Kelch in the fat, the recovery is almost not present, suggesting that Kelch knockdown results in smaller ring canals and also ring canals that do not allow for transport of cytoplasmic GFP. Okay. Right. So what are the physiological consequences of disrupting these ring canals? I told you that the fat body is a tissue that's very important for many aspects of development, but is also a tissue that endures a high basal stress. So when we looked at expression levels of our favorite stress reporter, which is the same one I showed you at the beginning of my presentation, we see that knocking down Kelch results in just higher basal levels of stress. But then in some outlier cases, we see extremely high levels of ER stress, but cells that appear to be paired to one another show disparate levels of stress reporter expression between paired cells. So this is the quantification for just overall stress reporter intensity across control and Kelch knockdown fat bodies, where the fat bodies are just generally more stressed. But then when I measure the difference in stress reporter expression between paired cells, we see a significant disparity in stress reporter expression, whereas steady state GFP expression is not really altered. So this leads me to tentatively conclude that the ring canals might play a role in buffering a stress response between sister cells, which are connected to each other through ring canals. So what about when we add additional stress? So in this experiment, these videos span 18 hours, where I do the same thing, dissect fat bodies, embed them in low melting agarose, and then image them over the course of 18 hours. And in both of these cases, this is control, and this is a Kelch depleted fat body. I've added a drug that induces additional ER stress, tunicamycin, and over the course of 18 hours, these fat bodies are actually pretty difficult to kill. You'll see maybe 2 or 3 cells die. And by die, I mean irreversibly lose GFP expression. However, when I knock down Kelch and then add tunicamycin, you see the lights go out. These fat bodies are extremely stressed and do not like having additional ER stress and are dying. So, right. So far, what I've told you is that we discovered that larval adipocytes in Drosophila possess ring canals and are paired to one another through ring canals, that they are present in all parts of the larval fat, in both males and females. Everything I showed you today was in females, but we also see similar things in males. The dimensions of these ring canals do not change while adipocytes themselves grow. These ring canals support the transfer of cytoplasmic proteins, ER-bound proteins, Golgi vesicles, but not mitochondria. And to our knowledge and to the extent of our experimental capabilities, not lipid droplets. And disruption of these ring canals leads to lower levels of intercellular transport of cytoplasmic protein, higher levels of basal stress, reduced cell survival. And I'm not going to have time to walk through this in detail, but we also see reduced serum protein secretion from the fat. But interestingly, no change in total triglyceride levels. And triglycerides are the major fat reserve in the fat body. We also see a nutrient-dependent developmental delay. So if there's one thing you take away from this talk, I would like for you to think of the ring canals and developing adipose tissue as transacting in the checking account of the fat, which is the proteins and the secretion, the things that the larva needs for active secretion as it grows. But the adipose acts at the adipocyte lipids act more like the savings account, where the lipids are stored for future use during metamorphosis and so are not actively required for ring canal mediated buffering or transport. And it's also stored and not necessarily actively transported between sister cells. So, oops, sorry, I forgot to show the data where we see reduced serum proteins and no change in lipid reserves when we disrupt ring canals. Okay, so in the last couple of minutes that I have, I just want to talk to you about something that's like consumed my thoughts over the past almost decade. So cells sharing, or 2 or more nuclei sharing some of the cytoplasm, appears to be a very conserved feature of several tissues. And similar to what I just showed you in the fat body, several of these tissues I'm showing you here also perform very important metabolic roles. Cardiomyocytes, adipocytes in vitro, lactating mammary cells, the exocrine pancreas are all, to some extent, have cells with multiple nuclei, 2 or 4 usually. And the liver is famously polyploid, with diploid, tetraploid, and octoploid cells, often containing 2, 4, or 8 nuclei as well. All of these examples from mammalian cells I'm showing you here arise from some variant of incomplete cytokinesis or truncated cell cycle. However, cell fusion can also give rise to binucleate cells, as is the case in the adult adipocytes in Drosophila. So Drosophila adipocytes really like to have multiple nuclei share a cytoplasm, however that comes to be. And so the function and outcomes of DNA or nuclear copy number and what it does for animal physiology is, something that I'm always very interested to talk about. So if you want to talk to me about it, I'm happy to be a friend or a colleague. And so I'd like to end my talk with a very brief overview of the research themes I'm interested in and what I hope to explore in the future as an independent investigator. And it all revolves around stress and resilience. As I mentioned earlier, another project I'm working on involves identifying and characterizing novel attenuators of the integrated stress response, which with some luck will be pre-printed very soon, I hope. Another thing that I'm interested in understanding is how persistent stress in metabolic tissues might influence their ploidy and how ploidy in turn might protect cells from the effects of such stress. To read more about how pervasive yet poorly understood this is, feel free to scan this QR code. And then further, I'm interested in how different levels of stress might influence different aspects of physiology. So there's research indicating that stress signaling might have several homeostatic functions. And I'm very, very interested in understanding how transient high stress might impact developmental transitions and also how pulsed and intermittent stress might influence biological oscillators. If you want to read more about homeostatic functions of stress signaling, there's a review that Lydia Grumai, my PI Deepika Vasudevan, and I wrote that just came out a couple months ago. Feel free to check it out. And with that, I would like to thank all the members of the Fly Stress Lab, past and present, who have contributed to this project and who've supported me. Big shout out to Flybase, which is under a lot of threat right now. So if you have some spare change lying around, please donate it to Flybase to keep it up and running. It's a resource that really fuels a lot of model organism research. And I would also like to thank the Center for Biological Imaging at Pitt that allows liberal use of all of their fancy microscopes. And with that, I'm happy to take any questions.
Right, thank you so much. I'll be reading questions from the Q&A as they come in.
Okay.
I'll start us off. So you made this incredible resource, the polyploidy resource, and I was wondering, based on your looking into where polyploidy exists in other organisms and other tissue and cell types, do you have any insights or thoughts about how this might have evolved?
That's a good question. I want to clarify that I did it as a graduate student while in the Botita lab, and most of it was curated by a fantastic then undergraduate student. I don't know specifically how polyploidy might have evolved, but I read somewhere that endopolyploidy, which is when multiple copies of the genome exist in one cell, has evolved more than 30 times throughout metazoan evolution. So I don't know how, because I'm not an evolutionary biologist, but I know it's happened multiple times, which means it's probably very important.
Yes, I was also referring to you gave multiple examples of polyploidy and specifically talking about the cases where they're sharing cytoplasm. So in the terms of just looking at the ring canal mechanism that you've been studying, I know you mentioned that there are other cases of this in other organisms, but can you speak more on that specifically?
Oh, sure. So I think it's been best studied as ring canals in the germline and as intercellular bridges in the germline of animals. But it's probably also been very well studied as plasmodesmata in plants, which is, so structurally, these are completely different because plants have cell walls and animals don't. But the root necessity for sharing cytoplasmic content seem to be to shuttle nutrients rapidly while still having like a nuclear control center within each cell. That's my hand wavy thought process, and I would love to study this more in somatic tissues where it also seems to be more prevalent. Yeah, I hope that sort of answers your question.
Yes, thank you. We also have another question from Celeste Berg.
Mhm.
Have you tried to manipulate embryo fat cells by altering cell cycle regulators to force complete cytokinesis?
Oh, that's a great question. I have not, although that is something I plan to do. One issue I anticipate with that is that because the fat body is so important and any manipulation of fat that early in development might result in a lot of developmental delay, we might not see animals make it to late larvae without significant defects. What I have seen is work from Max Frolov's lab has shown that DP mutants, so this is a mutant in a cell cycle master regulator transcription factor DP. DP mutants to a certain degree of penetrance have binucleate fat cells. And so this might be some form of completely bypassing an incomplete cytokinesis and cells. It's there's a phenotype for the opposite of what you're asking about, Celeste, is what I want to say. But I do want to do it. I haven't done it yet.
We have another question from Michelle Stars Gaiano. Nice work. I wondered if you looked for cytoskeletal elements running between the paired cells, suggesting regulated transport, or if you think the transport between them is all diffusion based.
Oh, that's a great question. So I tried very hard to super resolve in multiple colors. The fat body is very thick. It's about 50 to 80 microns thick, and the ring canals are less than a micron in dimension. So locating them has been hard. I have seen tubulin very close to ring canals, but I cannot conclusively say if we see cytoskeletal components traverse ring canals. However, I suspect that there might be some sort of a fusome or contiguous organelles that run through them that we would need to do some ultrastructure resolving to get at. Yeah.
Okay, Myler Lee asks, do you think there's a mechanistic link between the incomplete cytokinesis ring canals and polyploidy in the fat body cells? Does Kelch RNAi affect ploidy?
Kelch RNAi, I'm going to answer the second question first. From what I can see, the nuclear size does not appear to change. And so I don't think that Kelch RNAi affects ploidy, although there is a slight developmental delay. So animals might be acquiring their final ploidy much later. I think that the paper that I referred to earlier, Box et al. 2024, showed beautifully that the accessory gland of Drosophila undergoes sequential truncated cell cycles to give rise to cells that have ring canals that then binucleate and then endoreplicate and then incompletely replicate the genome. So some variant of like sequential truncation of the cell cycle is probably happening in the fat body as these cells differentiate and start making a lot of their secretory products and storing lipids. So I think that there is I think that the incomplete cytokinesis is step one of truncating multiple cell cycles, but that's all I have for you right now.
All right, I think that's all the time we have for questions, but feel free to reach out to the speaker if there were any questions you had that couldn't get addressed today. Thank you both, Dr. Colijn and Dr. Nandakumar, for your excellent talks. This seminar has been recorded and will be available on the SDB website next week. And please join us next month for next month's seminar on Friday, October 10th, when Dr. Austin Soroka from Fred Hutchinson Cancer Center and Dr. Heather Dingwall from the University of Pennsylvania will present. And thank you all for coming.